before I start with the presentation, I really want to acknowledge that this past month obviously has been very challenging for all of us and for the entire HD community. But despite the setback and the disappointment, we are really here today, Jeff, Kelly, and I, to reiterate and reaffirm that we are committed at WVE to this area. We are committed to the patients with HD, to their family, and to the entire community. And we'll today try to illustrate how we intend to continue our efforts to provide new promising treatment to this community. So what I want to do is to share the new clinical trials with SNP3, which is built on our belief that we need to have a very precise, targeted approach in lowering mutant huntingtin. And we want to be very specific because we want to reduce the toxic huntingtin, but we are aware of the important function of the healthy huntingtin that we really want to preserve. So that's our unique approach, and that's the leitmotif of the entire presentation, and I hope I can explain that well to you. So this new program also obviously benefits from the learning, the lesson that we have learned in the past, both good and bad, successes and failure. And it benefits from the much improved molecule that we have now that has a better pharmacology, better feature, that may address what were the limitations of the old molecules that, as you know, have failed to lower mutant huntingtin. So, oops. It's stuck. Voila. So this is just to remind you that I will have some forward-looking statements throughout this presentation. And for those of you who don't know WVE is a genetic medicine company that is focusing on the treatment of very serious diseases that are genetically defined. And so we build targeted approach based on that. And one of the conditions that we are treating is HD. And HD is in fact one of the most relevant therapeutic areas for us, and also one of the most advanced from a clinical development standpoint. Our approach to HD is certainly unique, as I tried to say before, and it's based on selectivity, so that we really aim at knocking down the mutant huntingtin protein while leaving the healthy one relatively intact. And our clinical program, WVE-003, we are about to announce, we don't have a name yet, but we are very close to disclose a name. So next time we'll meet, I'm sure we will have it. And we already started, we have submitted protocols, and we hope to dose the first patients this year. In addition to HD, also we have other therapeutic areas of interest, such as ALS, FTD, Duchenne, and those are also programs that are ongoing and in the early stages of clinical development. So our strategy to tackle HD is based on a few pillars. First of all, a strong scientific foundation that recognizes that healthy huntingtin has an essential role. It's a protein that is really important for the functioning of the brain. And second, we are really committed to provide a novel approach that is selective and that is trying to spare the healthy huntingtin, which I'll show you later has important roles in the brain. It's also based on our past experience and the learning that we have had from PRECISION-HD1 and 2, including being able to identify patients with this particular mutation much more quickly, including the ability of actually measuring the healthy huntingtin, which wasn't possible before. So we have put a lot of effort to try to improve the methodologies and to be able to enroll patients in the study very quickly and have pharmacodynamic markers, so markers that tell us that the drug is actually doing what it's supposed to do and what we think it's doing. And lastly, we can count now on a large clinical community that we have built throughout these years. We have qualified, experienced clinicians that have run our previous studies, and now they are all on board to run the SNP3 trial. So we hope that this time around, things can go much more quickly and much more efficiently. So how do we think of lowering this toxic mutant huntingtin, and what's our approach? This is based on two basic knowledges. One, that HD is not only a consequence of the presence of the toxic mutant huntingtin, but also a loss of function of the normal healthy huntingtin. And that the healthy protein, as I said before, carries out a number of critical functions for the brain, for the entire central nervous system. And as you can see in this graph, a healthy individual carries 2 copies of a gene that is responsible for the production of the huntingtin, so that this individual, when they are healthy, only produce healthy protein. But at the bottom, you can see that individual with HD will only have a single copy of the gene responsible to produce healthy huntingtin. So they will have the additional toxic effect of having unhealthy toxic protein, the mutant huntingtin. So why is the healthy huntingtin important? We know that the wild type, so-called wild type, is the healthy one. Huntingtin is essential for the functioning of the central nervous system because it's able to counteract various stressors that the brain encounters on a daily basis. It protects neurons against oxidative stress, protein misfolding stress, all those metabolic activities that occur in the brain, especially in those cells that are very highly active and have a high metabolic activity, they are prone to stress. And thanks to the healthy huntingtin, this stress can be counteracted, so it's preserved. It has a neuroprotective effect. In addition, it's important for the communication in the brain, right? So the so-called synapses, those are sites where neurons communicate with one another thanks to the trafficking of proteins that go from one to the other. Huntingtin protein actually helps that trafficking, so that communication across the billions of neurons that we have in the brain. And it also helps the so-called plasticity of the brain. Every time that we have connections in the brain, we form some permanent bonds that allows us, among everything, to form memories and to help cognition. So you can imagine how critical this protein is in the brain. And that's why we really want to preserve it with our approach. And now that you obviously, hopefully, I could convey the importance of the healthy huntingtin. What's important is that the understanding that HD is driven by these 2 factors, right? So we have, on one end, a gain of function of the mutant, the toxicity. On the other end, we have a loss of function of the very nice protective protein. And there is this ongoing struggle, as you can see in this cartoon, between the positive biological effect of the wild type huntingtin protein and the toxic effect of the mutated protein in the CNS. Unfortunately, people with HD, as we said before, already start at a disadvantage, right? Because they only have 50% of the good protein. And on top of that, they gradually lose ground to the disease progression. And if we knock down the wild type, that goes even faster. So we deplete the reservoir of healthy protein, which absolutely we don't want to do. So we believe that by preserving the healthy protein, we can achieve better efficacy. That could be a main driver of the efficacy of our drug, having this selective approach and preserving wild type. So how are we ensuring that this happens? We are using antisense oligonucleotides. Those are molecules that target the messenger RNA. And we are also using a second element, so-called SNPs, that I will talk about, which are kind of marker elements that allow us to recognize the mutated protein only, but they are not present on the healthy protein. So we'll use them as a, we used to say, a pin on the map, just to recognize where the mutated protein is so that we can attack it using the SNP itself. So as you may know, each protein in our body is produced starting from the information contained in the DNA. And that gets transferred to the RNA and then translated into a protein. And as you can see on the left-hand side graph, this is all the normal process that eventually brings us to the production of a healthy huntingtin. On the right-hand side in HD, your genetic information contains a mutation that is again transferred to the RNA, and then it's translated in the wrong protein. So that's where we intervene with the ASO, these oligonucleotides. Those are small, short stretches of DNA that complement the mutated RNA. And by attaching to it, they basically prevent the production of the mutated protein. So that's how we want to tackle it. So we want to stop the translation and transduction so that we don't produce that protein at all. The second element is the selectivity, as I said before, which is really important to our approach, right? And what allows us to be targeting specifically the mutated huntingtin gene is, again, the SNPs. These are called single nucleotide polymorphisms. And those are little variations, like single letter variations that happen throughout the DNA. And we have identified SNPs that are associated just with the mutant huntingtin gene, but not with the healthy gene. So we are able, with our oligonucleotides, to target that particular SNP and enable the prevention of production of the mutant huntingtin. So this is exactly what we are doing, both with the old molecules and the new molecules. I'll tell you how things have changed with the new molecules, but one thing that did not change is our aim at this selective approach, targeting the SNPs of interest just to knock down the mutant huntingtin. So we were able to identify 3 SNPs, and as you know, the SNP1 and the SNP2 were the target of our previous program with in HD, PRECISION-HD1 and PRECISION-HD2. And now the current program is focusing on the third SNP, the SNP3. Now, as you know, our previous candidates, SNP1 and 2, were evaluated in a phase 1/2 study. And unfortunately, the results do not really support further development of this program. They were presented at CHDI, and they will be presented again at HDSA. But just to summarize the results, we did not find any statistically significant reduction of the mutant huntingtin in either the core studies or the open label extension studies, both in PRECISION-HD1 and PRECISION-HD2. We have also seen, though, that these molecules did not cause any increase in inflammation and did not result in worsening of the disease progression. So despite this setback, obviously, we are immediately moving forward with the SNP3 program, which is estimated to be present, thanks to the SNP3, present in 40% of the HD patients, according to the current literature, but we are also doing some internal calculation to estimate what's the regional distribution and if these percentages are truly accurate. So our work with Huntington's disease continues with this program. And I want to highlight a few features that this new molecule has and the differences with the old one, the old molecules, SNP1 and SNP2. So first of all the development of the program is founded on the learning from the past and the scientific progresses that we have made internally in the company. One thing that obviously, and I want to keep saying this, didn't change is the allele selective approach. But wave 3, these new molecules, have been greatly improved from the previous SNP targeting candidate. It includes a new chemistry. So the backbones of these molecules has been changed in order to allow, thanks to new chemistry, to have a better potency, to have a better distribution so that the molecule goes in the parts of the brain where it's supposed to go. As you know, it's not easy to shoot a molecule in the deep brain. And thanks to this chemistry, we have found out that the molecule is better able to distribute in relevant central nervous system tissues. And the other characteristic, besides the potency and the better distribution, is the durability of the effect. So we were able to use preclinical models. We have seen that mice, which we didn't have with the previous molecules. We didn't have a mouse model where we could really ascertain that our molecules knock down the mutant protein. In this case, we do have it. We have both in vitro and in vivo models where we could see the pharmacology and the pharmacodynamic effect of the molecule. And we have also, thanks to all this knowledge from the animals, been able to design a study that is adaptive. It's a little bit smarter than a fixed dose, fixed regimen as we have done in the past. But thanks to this adaptability, we may choose better doses that may guarantee better safety and better proof of concept, proof of biology, and maybe much more rapidly. So we have also greatly improved the way we are identifying patients, because don't forget, we need to ascertain that they have a mutation, and we need to ascertain that they have the SNP of interest, this SNP3. And that wasn't an easy endeavor. It took us some time, more than a couple of months. Now we can do that in 2 weeks. We have greatly improved this methodology so that as soon as the sample is sent to the lab, it can come back with the results in 2 weeks, and patients will know if they are eligible for the study. So just to show you some of the results, which are quite encouraging, that we have seen both in vivo and in vitro. And this is really the foundation, and this is why we are so excited about moving forward with this program. You can see that there is in vitro a potent effect of the SNP3 as compared to another compound that doesn't have any specificity, knocks down both good and bad protein. In our case, we can show that even if we have the same potency, our approach is specific and knocks down just mutant.
Sorry, Vissia, sorry, in vitro is cells in the laboratory, huh?
Yes. Oh, sorry. Yes. Did I slip into scientific language? Yes.
A little bit
so
there.
Both in the Petri dish and in actual animal models that resemble the HD disease, basically, we could see that this approach was selective, meaning that the good protein stayed and the bad protein was knocked down. And you can see it here in the right-hand side. The striatum is a part of the brain that is really relevant for this disease. And you can see that we were able to knock it down over 75%, and that this effect was really had a long duration over 12 weeks. So we are very encouraged by this fact that will allow potentially to dose less frequently if we realize that higher doses of this drug have a sustained effect, then we can dose patients much less frequently, which is always in our mind, obviously. So we have a potent and durable effect. And we have demonstrated selectivity both in the plate, in the lab, and in the mouse. So we were able to incorporate all these lessons learned, right, in our clinical trial with NIP3. And leveraging all this data allowed us, so first of all, to be able to genotype. This is very sensitive. I don't know what's happening. We were able to implement a few things. So first of all, learning from the animal model was really important, because differently from SNP1 and 2, where we didn't have an animal model, we had to start with a dose that was not toxic, obviously, but maybe it wasn't the optimal dose to determine an effect in vivo. In this case, we could leverage all this in vitro and in vivo data and predict a human dose that is potentially efficacious. So it is at least pharmacologically active. And from there, we can move up and have better doses and better dosing frequency and optimize the treatment, both for efficacy and for safety. And we are implementing, obviously, the genotyping assay for rapid screening of our patients, as opposed to the lengthy processes of the past. And again, we can count, obviously, on very qualified clinicians that have been working with us very collaboratively. This trial, as you see in the right-hand side, incorporates all that, and it's a single ascending dose and a multiple ascending dose study all in one, where patients come in, get the first dose, then there will be a period of washout, and then they get additional doses. And all the information coming from this lower cohort will inform, thanks to a safety committee and a dose escalation committee, what's the best dose to give next. So they will ascertain pharmacokinetic, they will ascertain safety data, possibly, if we're able to do that, also pharmacodynamic data, so measuring the protein itself, in order to optimize the doses for the next level. Again, the primary objective of the trial is safety and tolerability, but we will put a lot of effort in evaluating the pharmacokinetic and pharmacodynamic effects. So we look at marker of efficacy. We want to see, we will measure mutant huntingtin to see if it's knocked down. We will measure wild type huntingtin, the good one, to see that it's preserved, basically. And we will measure all the markers of inflammation and neurodegeneration to show that our drug does not cause toxicity in the brain. This trial will enroll around 40 patients, and then patients obviously have to have a confirmed diagnosis of HD. And we are trying to get the earliest stages of HD, but it's still stage 1 and 2 will be included. And there will be clinical parameters that will determine inclusion and exclusion in the study. But we are trying to be as open as possible to include most of the patients. And most of the patients that have also participated in PRECISION-HD1 and PRECISION-HD2, because we have done assessment of the pharmacology of those molecules. And now we know that the washout period is quite short, so those patients can screen for this new study if they desire to do so. The age is similar to other studies, between 18 and 60 years old. And we are already, as I said before, in the process of activating sites all over Europe. And we are hoping to include more countries and more sites in the next few months. And again, we really hope to have the first patient in very shortly this year. Again, just to summarize, unfortunately, the PRECISION-HD1 and 2 trials are being discontinued. So all our patients are undergoing the last visit at the moment, and we are collecting as much information as possible because that information is really valuable to us. So, and patients have been so kind to provide CSF so that we can do one last assessment. They are undergoing MRI, they are undergoing all the safety assessments, and then they can obviously screen for the new study. As I said, our new candidate is ready to go. We have got approvals from a few countries already, both on this side of the ocean and your side of the ocean. And we hope to capture as many patients as possible that were already enrolled in the other studies. We are still excluding, obviously, patients who were on active treatment with tominersen, but if patients were on placebo, obviously in that phase 3 study, they are also eligible to screen and then potentially enter the study. So I just want to reiterate my gratitude to all the patients and the community that has been so attentive and so important for us to finish those 2 studies and to start this new SNP3 study, which gives us a lot of hope. Again, all the associations that Jeff and Kelly have established relationship with have been extremely, extremely helpful, and obviously all the patients and their family. Thank you so much. So I'll be happy to address any questions you may have.